Hi, and welcome to Week 3 of Applied Immunology. This is Lecture 3A, which starts the Week 3 module, where we'll finish learning about functions of the innate immune system. Today we'll talk about how innate immune cells take ingested particles from microbes, which are proteins called antigens, and process or prepare them in order to stimulate adaptive immunity. We'll briefly cover the different types of professional antigen-presenting cells, or APCs, the categories of antigen presentation molecules used by APCs, which are called major histocompatibility complexes, or MHC. And lastly, we will discuss the intracellular processes that allow APCs to chop up a pathogen into pieces that can then be recognized by the adaptive immune system. If you remember back to our introductory lectures in Week 1, we learned that the adaptive immune system consists of B and T lymphocytes, or B cells and T cells, which can recognize specific protein sequences derived from pathogens, which we call antigens. B cells and T cells each express lymphocyte antigen receptors that enable high specificity binding to peptide fragments of these pathogen-specific antigens. We learned that these receptors on B cells are called B cell receptors, or BCRs, which in their soluble form are antibodies, while on T cells they are called T cell receptors, or TCRs. Aside from the obvious naming differences, these antigen receptors also differ with respect to their structure and function. The BCR can bind to soluble antigen and does not require antigen-presenting cells, while the TCR can only recognize foreign peptide sequences when they are presented while bound to a major histocompatibility complex, or Mhc molecule, which is expressed by antigen-presenting cells. So for our lectures that deal with antigen processing and presentation this week, keep in mind that these things are required to activate T cells of the adaptive immune system, but not B cells. Before we get started with some more specifics today, I want to outline the two main goals, or outcomes, that the immune system tries to achieve through the processes of antigen processing and presentation. Number one is to take a chunk of protein, called an antigen, which belongs to a pathogen that has either been phagocytosed or is actively infecting a cell. This antigen is then chopped up or processed into smaller pieces called peptide fragments that can be loaded onto these Mhc proteins expressed by APCs so that the peptide sequence can then be recognized by a T cell receptor. Later on, we'll learn that the specific peptide fragment that dominates T cell responses to a certain pathogen is often referred to as an immunodominant epitope of that given pathogen. So if we refer to peptide fragments or epitopes, they're usually referring to a similar thing. Number two is to only bind to TCRs that are expressed by T cells that are best suited to eliminate that same pathogen. And by this, I'm referring to the fact that there are different types of T cells that are activated by different types of Mhc molecules that are expressed by certain types of cells. And that these different categories of T cells deploy distinct effector mechanisms which promote pathogen clearance. We've been introduced to the two major subtypes of T cells before. There are helper T cells, which express the surface marker CD4, and then there are cytotoxic T cells, which express the surface marker CD8. So depending on where an antigen-presenting cell encountered a given antigen, this can influence the type of Mhc molecule that its peptide is presented on, which therefore determines whether CD4 or CD8 T cells can be activated in response to that pathogen. These goals of antigen presentation, not surprisingly, are accomplished in large part by professional antigen-presenting cells, or APCs. These include macrophages, which we've discussed before when covering professional phagocytes, as well as dendritic cells, whose primary function is antigen presentation. Dendritic cells phagocytose small amounts of cellular debris specifically for the purpose of antigen presentation. It's worth noting that macrophages and dendritic cells are located in peripheral tissues, but upon their activation by stimuli associated with infections, such as PAMPs and cytokines, they can migrate to the lymph node or spleen, the secondary lymphoid organs, where they can initiate immune priming. Lastly, B cells are also professional antigen-presenting cells, although this is done in addition to their primary functions, which involve secreting antibodies and forming long-lived memory B cells. B cells aren't actually phagocytes, but they can internalize antigens that are bound to their B cell receptor, which in turn are then presented to T cells. Unlike DCs and macrophages, B cells are primarily located in secondary lymphoid organs such as the spleen and lymph node, and they're also present in circulation in the blood. For this course, when we talk about antigen presentation, we'll kind of primarily be talking about this being done by innate immune cells like macrophages and DCs rather than B cells, but just keep in mind that this lymphocyte population is also capable of activating T cells in certain contexts. Now, what exactly defines these three cell types as being able to function as professional antigen-presenting cells? This simply refers to the fact that these cells can present antigens on both types of MHC molecules, which are Mhc Class 1, or Mhc1, and Mhc Class 2, or Mhc2. These two categories of MHC differ with respect to a number of key properties, but for starters, I want to first emphasize how these two MHC molecules differ in terms of their distribution of expression amongst cell types, as well as which subsets of T cells they can activate. First, on the left here, Mhc1 is expressed by all somatic cells, which encompass both immune and non-immune cells, with the exception of red blood cells, which don't express any sort of antigen presentation machinery. All professional antigen-presenting cells also express Mhc Class 1, and peptide presented on Mhc1 can only activate cytotoxic CD8-positive T cells, which are important for performing cytolytic effector functions. This is in contrast to Mhc Class 2 on the right, which is expressed only by the professional APCs depicted here. Also, peptide presented on Mhc Class 2 can only activate helper CD4-positive T cells. So in general, just please remember to associate Mhc1 with CD8 T cells, and then to match Mhc2 with CD4 T cells. Another key way in which Mhc molecules differ is in the cellular compartments from which they can report antigen from, and how this is linked to the downstream effector mechanisms employed by T cells once they become activated in response to peptide Mhc. Mhc Class 1, which again is expressed by most somatic cells, can load pathogen-derived peptides that are sampled from the host cell cytosol. Again, Mhc1 activates cytotoxic CD8 T cells, whose main effector function is to deploy cytotoxic mechanisms that kill the cell which is presenting antigen. And this makes logical sense when you consider that these cells have pathogen-derived proteins present in their cytosol, which really only occurs in a case of active infection with a virus or intracellular bacterium. In this situation, you would want to kill and eliminate the infected cell in order to get rid of the survival niche that's required for that intracellular pathogen. Professional APCs present antigen on Mhc Class 2 in addition to Class 1. And for Class 2, they can source these antigens from distinct compartments compared to Mhc Class 1. One is the vesicular compartment, which refers to many membrane-bound organelles within a cell, including those derived from the endoplasmic reticulum in the Golgi complex. Importantly, this includes things like endosomes and phagolysosomes. There are numerous types of bacteria and parasites that can survive intracellularly by kind of walling themselves off in endocytic organelles where they can evade the host immune response, which we'll refer to as intravesicular pathogens. And the vesicular system also contains extracellular pathogens that have been phagocytosed by macrophages or dendritic cells. Therefore, the vesicular compartments can also sample pathogens that have been taken in from the extracellular space. Notably, the extracellular space can also be sampled by B cells following the internalization of BCR antigen complexes, as shown here on the right. But in either one of these cases, MHC2 enables antigen presentation to helper CD4 positive T cells, which have different effector functions compared to cytotoxic CD8 positive T cells. These functions include the activation of non-cytolytic mechanisms to kill intravesicular pathogens in the center here, or in the case of antigen presented by B cells, helper T cells can induce the secretion of antibodies, which are also referred to as immunoglobulins or IG, that are secreted by B cells to help opsonize and eliminate pathogen targets. Note that in the case of professional APCs that present antigen on MHC2, antigens sampled from these compartments can be derived from extracellular particles that are not necessarily indicative of active infection in the MHC2 expressing cells. So the downstream effector mechanisms engaged by CD4 T cells don't typically lead to the killing of the antigen-presenting cell that was responsible for presenting antigen in the first place. We'll learn a lot more about the effector functions of CD4 and CD8 T cells next week, but for today I want you to focus on the fact that because professional APCs can present antigen on MHC2 in the absence of their own intrinsic infection, they can lead to distinct forms of T cell activation compared to MHC1 expressing somatic cells. Let's begin to learn more about the specific structure of Mhc molecules, starting with Mhc Class 1, which again is expressed by most somatic cells. Both Mhc1 and Mhc2 molecules are expressed on the plasma membrane and share a similar three-dimensional structure, where they consist of 4 protein subunits whose binding region kind of looks like a hot dog bun. As you can see at the top of this molecule here, there are 2 ridges on the side, which make up the bun portion in this analogy, with a groove in the middle called the peptide binding cleft. In this analogy, the foreign peptide would be what makes up the hot dog. Note that the specific subunits used to make up MHC1 differ from those used to make up MHC2. and that the subunits used to make MHC1 interact with the co-receptor CD8, which is expressed by cytotoxic T cells. Now, if we take this molecule on the upper left and imagine rotating it 90 degrees towards you, then we have a view of what the MHC molecules look like from their top, where we're looking down into the groove of the binding cleft. This schematic on the lower left gives us a structural view of the molecule, where you can see that the binding cleft is a beta sheet that's made up of alpha-1 and alpha-2 subunits of MHC1, and that this beta sheet is separated by two ridges, or the buns of MHC1, which consist of alpha helices. Now, the diagram in the center shows the protein structure of MHC1 in yellow again, this time along with a peptide in red that has been loaded into the binding cleft. And so by this point, we know that MHC1 presents cytosolic peptides, but how exactly are these peptides generated for binding? The answer is the proteasome, which is a proteolytic complex found in the cytosol of all cells. The proteasome here in purple forms kind of a tube-like structure, which binds to polyubiquitinated proteins in red found in the cytosol. So note that ubiquitinylation is a post-translational protein modification that's used to tag old or unneeded proteins for destruction in a cell. Once the proteasome binds to ubiquitinated proteins, it feeds them through the center of the proteasome tube where proteolysis occurs. This degrades the protein into shorter peptide fragments, which are then spit out of the opposite end of the proteasome tube. The proteasome is important for the process of normal protein turnover in healthy cells, so it degrades a lot of self-proteins in addition to any pathogen-derived proteins that are also floating around in the cytosol. These pathogen-derived peptides are the ones that actually load onto MHC1 for antigen presentation. As a side note, exposure to interferons can also induce expression of certain subunits of the immunoproteasome, which allow this complex to more efficiently generate proteins that are the appropriate size for binding to Mhc Class 1. MHC1 also has some specific requirements for the types of peptides that can be loaded. These fragments must be 8 to 10 amino acids in length, which is important to keep consistent because the N and C termini of the peptide actually contribute to its binding in the binding cleft. The binding cleft is also a closed groove. Lastly, it's worth noting that the MHC1 molecule is a relatively unstable protein when it exists without a peptide loaded into the binding groove. So in the absence of foreign peptide, the MHC1 molecule requires interactions with stabilization proteins called chaperones, which we'll discuss next. Now that we know more about the structure of MHC1, let's look at where MHC1 is assembled and some of the mechanisms required for its stabilization and peptide loading. First, MHC1 is generated and held in the endoplasmic reticulum, or ER. During its formation, partially formed MHC1 is first stabilized by a chaperone called calnexin, which allows for the recruitment of the beta-2 microglobulin subunit and the assembly of all 4 subunits of MHC1. Immature MHC1 molecules are extremely unstable until complexed with this beta-2 microglobulin subunit, but even once they're fully assembled, still exhibit a degree of instability until peptide is bound. Once fully mature MHC1 is assembled, it then binds to a complex of chaperone proteins, which include calreticulin as well as ERP57. In addition to stabilizing the MHC1 complex, these chaperones also hold the binding cleft in an open conformation so that the peptide can be loaded. So at this point, MHC1 is fully formed, but it still needs to bind foreign peptides. So it's worth noting right now that the binding region of this yellow MHC1 molecule faces towards the interior or the lumen of the ER and is separated from the cytosol by the ER membrane. But we also just learned that foreign peptides for MHC1 are generated by the proteasome that's present in the cytosol. So how do these peptides actually meet up with the MHC1 binding cleft? The answer is this green complex here called TAP, which stands for transporters associated with antigen processing. TAP is obviously located at the ER membrane and functions as a transporter channel that takes these little red peptide molecules from the cytosol and translocates them into the lumen of the ER. Once delivered into the ER, these peptides can be further processed by a peptidase called endoplasmic reticulum associated peptidase, or ERAP, which trims peptides over 10 amino acids long until they fit the range that's required to fit into the MHC1 binding cleft. This process yields peptide fragments that are within 8 to 10 amino acids long and are finally ready for loading into the binding cleft of MHC1. Upon peptide binding, the MHC1 complex is fully stabilized and can then be transported from the ER to the plasma membrane, where it is now ready for antigen presentation, which we'll discuss in the next lecture. Next, let's learn about the second category of MHC molecules, which are Mhc Class 2. Again, these are expressed strictly by professional antigen-presenting cells. The 3D structure of MHC2 is similar to MHC1 in that it forms a hot dog bun that can load foreign peptide in the groove of the binding cleft, although the specific subunits that make up MHC2 are different from those in MHC1. Note that because these subunits are different, MHC2 can bind to the co-receptor CD4 instead of CD8, which allows it to present antigen to CD4-expressing T helper cells. At the center here, this diagram shows the protein structure of MHC2 in yellow and green, along with a peptide in red, which has been loaded into the binding cleft. MHC2 has some key differences compared to MHC1 in its binding cleft. First, rather than only binding to a specific length of foreign peptides, Mhc Class 2 is unrestricted in that it can bind to fragments that are 13 amino acids or longer. Because there are no restrictions on the exact length of the peptide, neither terminus of the peptide are involved in the actual interactions with Mhc Class 2. And unlike MHC1, MHC2 has an open binding groove, but similarly to MHC2 is also quite unstable in the absence of foreign peptide loaded into the groove of its binding cleft. As MHC2 can sample the vesicular system of a host cell, peptides are drawn in from vesicle-bound organelles, including endosomes or phagolysosomes. Importantly, these organelles must be acidified, as the endosomal proteases called cathepsins, which are responsible for generating MHC2-compatible peptides, are inactive at a neutral pH. So, phagocytosed or endocytosed proteins from the extracellular space must meet up with something like a lysosome, which has an acidic pH. And this activates the cathepsins, which are these little blue molecules in the diagram, which are responsible for chewing up a larger foreign protein into peptide fragments, which can then be loaded onto MHC2. Now that we've established the structure of MHC2, let's get into how peptide is loaded onto this complex. First, like MHC1, the subunits of MHC2 are assembled in the lumen of the endoplasmic reticulum. Also similarly to MHC1, MHC2 is unstable when not bound to peptide, so it requires stabilization with a protein called the invariant chain, which is abbreviated as uppercase I and a lowercase I, or II. The invariant chain stabilizes MHC2, but also it importantly blocks the loading of ER peptides into the MHC2 binding groove. So remember that the TAP complex brings peptides from the cytosol into the ER, which if they were bound to MHC2 means that it would be presenting peptides from the cytosol, which would require a very different type of T cell response. So in order to maintain the ability to only respond to antigens encountered in the vesicular system, MHC2 must exclude binding to cytosolic proteins, which is accomplished through the invariant chain, blocking its peptide binding groove, specifically in the ER. Once assembled, the complex of MHC2 bound to the invariant chain is then exported from the ER to endosomes, which float around in the cytoplasm. The next important step for MHC2 is when the endosome encounters an acidic environment, such as when it fuses with a lysosome. Again, the acidic pH in this organelle is responsible for activating proteases called cathepsins, which upon activation will cleave the invariant chain into little pieces. Importantly, almost all of the invariant chain is degraded upon acidification, with the important exception of this small purple fragment, which is still attached to the peptide binding groove of MHC. This fragment is called CLIP, which stands for Class 2 Associated Invariant Chain Peptide. So although most of the invariant chain has been removed, at this point CLIP still blocks peptides from interacting with the binding region of Mhc Class 2. This acidified endosome can also contain foreign proteins, which are cleaved into peptide fragments by cathepsins again, although CLIP prevents their loading onto MHC2. So what actually needs to happen in order to replace CLIP with a foreign peptide? The answer is this green and teal protein in the right panel called HLA-DM. HLA-DM has a similar structure to MHC2, and when it associates with MHC2 in acidified endosomes, it induces a conformational change that allows MHC2 to release CLIP from its binding groove, and this frees up the site. The binding with HLA-DM holds it in an open conformation, which allows for Mhc Class 2 to bind to a foreign peptide instead. Again, like we saw with Mhc Class 1, the successful loading of peptide onto MHC2 stabilizes the entire complex, and this allows for MHC2 to separate from HLA-DM and then traffic to the plasma membrane, where it can then interact with helper CD4 T cells through the process of antigen presentation. Lastly, for today, I just want to bring up one important exception to the rules of antigen processing that we've covered so far in this lecture, which is something called cross-presentation. At the beginning of this talk, we established that MHC1 can present cytosolic peptides associated with intracellular infection to cytotoxic CD8 positive T lymphocytes, while MHC2, that's present on professional APCs, can present internalized or vesicular peptides associated with extracellular infection to helper CD4 T lymphocytes. However, there's one special process seen only in dendritic cells, where exogenous or extracellular antigens can be taken up and then processed for presentation on Mhc Class 1. This process is called cross-presentation and is a specialized mechanism where extracellular proteins that are taken up by endocytosis or phagocytosis are shuttled out of these endocytic compartments and instead transferred into the ER, where they can then be processed for presentation on Mhc Class 1. So why does this specialized mechanism exist if you consider the fact that most cells already express MHC1 that can activate CD8 T cells? And the answer has to do with immune priming, or the process through which antigen is presented to naive T cells in the secondary lymphoid organs, such as a lymph node. Although most cells express MHC1, non-professional APCs actually lack the ability to migrate to the lymph node from their tissue of residence. So they can only stimulate CD8 T cells that have already been activated and migrated to the site of infection. DCs, however, are specialized in that they can actually pick up antigen from a peripheral site, then enter the lymphatics and migrate to a lymph node where they can then initiate immune priming through the activation of naive T cells. Although DCs themselves may be infected by an intracellular pathogen that would result in traditional antigen presentation through MHC1, the likelihood of that infected cell actually maintaining its functionality and surviving long enough to travel to the lymph node and find a T cell capable of recognizing that pathogen is pretty unlikely. So cross-presentation is extremely important in that it allows dendritic cells to phagocytose dead cell debris that are resulting from infection, which likely includes pathogen-derived protein antigens, and then take these antigens and travel to the lymph node to present them to naive CD8 T cells in order to initiate cytotoxic adaptive immune responses. We'll learn more about the process of immune priming and T cell effector functions in week 4. But keep in mind that cross-presentation by dendritic cells is an important alternative mechanism of antigen processing that goes against some of the general rules that we've established today. To summarize, today we've learned that antigen processing within both immune and non-immune cells allows them to prepare proteins derived from pathogens in such a way that they can be presented to T cells in order to initiate antigen-specific adaptive immunity. We've also learned that peptides can be processed and presented on 2 categories of major histocompatibility complex, or MHC, which are Mhc Class 1 and Mhc Class 2. These 2 different MHCs present antigen from distinct cellular compartments, which determines the types of T cells that can be activated in response to antigen presentation. And also that these responses are tailored to fit the types of infections associated with the antigen taken from these different cellular compartments. We've also learned how the expression of MHC1 and MHC2 differ among cell types. And all of these major concepts are outlined in the bullet points listed on this slide. That's it for today's lecture, but please remember to watch the remaining 2 lectures for week 3, which will cover antigen presentation, as well as lymphoid cells that have innate immune functions.